Hi y'all, this is Dr. White. We're going to go over this week's cell bio topic, which is membranes, biological membranes. But first we're going to start with a little bit of a review. There is a follow-along assignment in iClicker, so go ahead and open that up and get ready to answer those questions. And let's get started. So, come on now. Sometimes the arrows just don't want to work. Okay, so we're going to review a little bit. First, we've got our characteristics of life. So, some characteristics of life. Life is structured, ordered and structured. Life can reproduce itself. It grows. It takes in and utilizes energy. It exhibits homeostasis and responds to its environment, particularly by sensing external stimuli. And life evolves. These were taken pretty much directly from nasa.gov. So if you want to read the full text, you can head to that link. Okay, so go ahead and answer your iClicker question here. This was intended to be a multiple answer question. I have since learned that you cannot do, that's not going to work, at least not right now. So just pick any correct answer for this one in iClicker. So select an answer that is an example of a cell exhibiting a characteristic of life, right? So we've got cyanobacteria divide, increasing their culture density to logarithmic phase. B, a dictyostelium cell forms a fruiting body to distribute spores. C, a prion denatures proteins in a host organism. D, moss cells maintain water levels through osmosis. Or E, mycelium absorbing nutrients from decaying organic matter around it. So let's break this down a little bit. Cyanobacteria divide, right? Division is a form of asexual reproduction. So this is characteristic 2. Okay, dictyostelium cell forms a fruiting body to distribute spores. This is another method of reproduction. That's characteristic 2. We'll come back to C. Moss cells maintaining water levels through osmosis. Osmosis and maintaining, that is homeostasis, right? So that is life trait number 5. And mycelium absorbing nutrients, that is number 4 here, taking in and utilizing energy. So number 4. So the distractor here is C. So a prion denaturing proteins in a host organism. Prions are a type of protein. So this is like a protein gone rogue, proteins that can denature other proteins, and then it becomes like a disease in the way that it develops in an organism. But a prion is not a cell, right? So it's not like a bacteria where it infects and each bacterium is a cell. A prion is just a protein. So this is not an example of a cell exhibiting a trait of life. Okay, so let's go over our types of cells. So we've got, in general, we got our eukaryotes and our prokaryotes. And the big feature between them is the nucleus and the membrane around it. So a membrane-bound nucleus and membrane-bound organelles. That's the big thing about eukaryotes, is that there is internal membrane systems in place. Prokaryotes have a nucleoid region where their DNA is localized, but it is not enclosed in a membrane. They also have circular chromosomes. Now, both have plasma membranes surrounding their cells, both have cytoplasm, and both have ribosomes. Okay, so true or false, no prokaryotes have internal membranes. Pause the video to think about that if you like. And would you look at this? These are my TEM images of cyanobacterial cells from my dissertation. And so cyanobacteria are photosynthetic bacteria, and because they are photosynthetic, they have to have a place to conduct photosynthesis. They have internal membranes. So each of these dark lines is called a thylakoid membrane. All around the cells, and over here as well. So internal membranes. Prokaryotes can have internal membranes, but that is definitely the exception to the rule. Okay, so let's go over our biomolecules. Answer this question real quick. It is the 4 categories of biomolecules. Okay, now biomolecules are typically polymers. They are molecules made up of smaller pieces. So polymers made of monomers. So for proteins, the monomer is what? DNA, the monomer is what? And then polysaccharides, the monomer is what? Okay, so go ahead and answer that question here. So this one, the leftmost would correspond to the top blank, and the rightmost would correspond to the bottom blank. Okay? Okay, so now we're going to learn about biological membranes. The key things I want you to take away from this are to be able to describe the primary features of biological membranes and explain how those structural features can relate to the functionality of a membrane. And then I want you to be able to connect those functions of the membrane to our characteristics of life. And I want you to be able to predict whether a molecule will be able to pass through the membrane based on what you know about molecules and about membranes. Okay? So these are our primary features of biological membranes. Membranes are bilayers made up of lipids and proteins. They are selectively permeable, they are fluid, and they are asymmetric. So let's talk about that first part, the bilayer. So, are we over here now? Yes, we are. So now I'm going to be drawing in my little paint window at the bottom. So biological membranes are made up of 2 layers. You may also know this as a phospholipid bilayer, but they are not exactly the same thing. A phospholipid bilayer would be just phospholipids. A biological membrane has a lot of other components, but the key thing is our phospholipids, right? And they are present in 2 leaflets, okay? So the head of the phospholipid is going to be facing either side, either outside of the cell or towards the cytoplasm. So this would be the external facing leaflet, and this would be the cytosolic facing leaflet. The cytosol facing leaflet or the cytosolic leaflet. Cytosolic leaflet. Okay, this internal space is where those hydrocarbon tails are located, right? And so those hydrocarbon tails are very nonpolar. So this region, wow, that was just a terrible bracket. There we go. This is hydrophobic. Okay, so now phospholipid bilayers can form, we can look at them as sort of like flat examples, like you can see in that top portion, or we can see them in spheres like liposomes. Vesicles are also made up of biological membranes. Now, phospholipid bilayers or biological membranes, they have the capacity to grow and reshape, right? And to be able to grow and reshape, they need to have more lipids added to them. But lipids are only added to one side of the membrane. So if you think about it, the cell can't add a lipid from outside, right? It has to be added on the cytosolic side. And because of that, if we think about this, if we've got our outside layer and our inside layer, right? And we keep adding to our inside layer. Eventually, our inside layer would get so big that it would catastrophically fail, right? So that's bad. We don't want that. So there are these two types of enzymes that help with this particular issue. So the first is called a scramblase. Scramblases are nonspecific, and their whole job is just to make sure that that strain is alleviated. Okay, so they're just going to take things from one side, switch them to the other, so that the two leaflets of the membrane are mostly equal. Flipases are specific. So we can see here, we got our scramblases. They're just moving things around willy-nilly. Our flipases are going to be moving things very specifically. So if you think about membranes, different membranes have different jobs. The Golgi membrane has a different job than the ER membrane. The cell membrane around the entire cell has different jobs than the internal membranes, right? So these flipases, their whole job is to take the lipids that need to be in one place and move them there, right? So this is one of your iClicker questions. I want you to think about why we would need an enzyme to make these adjustments to the membrane. Why do we need an enzyme to transfer phospholipids from one leaflet of the membrane to the other leaflet of the membrane? Okay. Now, we just mentioned this with the flipase and its specificity. So the two sides of the membrane are not mirror images. So the inside of the membrane is not the same as the outside of the membrane. The different faces of the membrane have different jobs. So if you think about cell signaling, we have, well, let me switch over to here where I have some more space. Okay, so if we have our cell, right, and our cell needs to be able, we'll do it like this, make a kinase. And our cell needs to be able to conduct signaling and sensory processes. So these kinases, for one, they're not going to be in all of the organelles, right? They're going to be located on the cell membrane to sense external stresses. And then any lipids involved in those stress responses would need to be in this area, either on the outer leaflet or the inner leaflet, right? So that outside leaflet is going to be different. It's going to have different signaling proteins than the inside. And the two sides of the Golgi are going to be different. The interior and exterior of different vesicles, those are going to be asymmetric. What's inside the vesicle is going to have different needs than what's on the outside of the vesicle. Okay. Let's flip, and this is our second to last. I think this is our, nope, second to last trait, membrane fluidity. So phospholipid bilayers and cell membranes in particular are fluid, right? They are not static. The membrane shifts, it moves, the phospholipids spin. And this fluidity is essential to the function of the cell. So this is essential for growth and movement. Those phospholipids have to be able to shift in order for the cell to get larger and eventually divide, to be able to transfer molecules into and out of the cell. So phagocytosing other extracellular nutrients or signal molecules or vesicles moving within the cell need to be able to form. So that requires membrane fluidity. Signal cascades, those require membrane fluidity because the proteins that conduct those signal cascades are spanning the membrane. So the fluidity, the movement of the membrane is going to affect how those signaling proteins operate. And responses to stress like cold temperatures. So membrane fluidity is affected by different factors. We got temperature, we got cholesterol, and we got saturated and unsaturated fats. Okay, the first is cold versus hot. So if we think about this cold temperature, right? Our phospholipids are going to pack really tightly together. There's less energy for them to be moving. They're going to be very close together. And if they are clustered very close together, there's less space for movement too. So this is going to be more rigid. Okay, now hot temperatures allow more space. There's more energy for movement. There's more space for movement. So a hot temperature membrane is going to be more fluid. Okay, and this is all considering that this is just phospholipids, but it's not. We also have cholesterol. Cholesterol is that other category of lipid made of ring structures instead of hydrocarbon tails. So we've got our cholesterol molecules here circled in pink, and cholesterol is pretty sporadically found throughout the membrane, and it acts sort of like a buffer. So if we've got our membrane here, and I don't want to draw a cholesterol molecule, so I'm going to put it in a different color in a second. Draw our little phospholipids here first, and we'll put our cholesterol in green. So our cholesterol, we're just going to make it like a little diamond. It's in between these phospholipids, right? So if the phospholipids are trying to like cluster, get closer together, they're not going to be able to. That cholesterol is going to keep them from squishing too closely together. But cholesterol also works in the opposite way. Remember, it has that little bit of polarity on the end, our little, what is it? It's our oxygen, but on the end of the cholesterol. So we got a little bit of polarity. And so our polar portions of our phospholipids are kind of drawn to the cholesterol too. So not only is it keeping them from crushing together, it's also keeping them from pulling apart, right? So in cold temperatures, the cholesterol keeps it from getting as rigid. And in hot temperatures, it keeps it from getting as fluid, right? So it helps keep our membrane fluidity a little bit more even, okay? And our last factor here is saturated versus unsaturated fats. This is one that you are probably familiar with. So a saturated fatty acid is one that is full of carbons, saturated with hydrogens, right? So carbon can have 4 bonds. 2 of its bonds are going to be taken up by the other carbons, and then it's going to have hydrogens sticking off, okay? So when we have all of these extra spaces filled by hydrogen, it's all single bonds, right? So, and then there's only one way that those single bonds can shape themselves, and that's this very sort of linear, shaped tail. The other way that we can see the formation is in an unsaturated fatty acid. So in this situation, it is not full of hydrogens. So this carbon right here, it has one hydrogen, but it has formed a double bond with another carbon. And those double bonds can have directionality, so they can either be cis or trans, right? So the carbons are going to have an angle to them in that bond. So we get these bent fatty acid tails. So if we have some phospholipids that have these very linear tails and some phospholipids that have these bent tails, we're going to get different membrane fluidities. So think about this for a second. If we have more of these linear tails, would the fluidity be higher, lower, or the same, right? Think about how if you have a bunch of pencils in a bundle, it's very easy to pack them in really tight. But if you were to have like twisty ties, something that bends, it'd be a lot harder to have them sort of in an organized bundle. So you can think about it kind of like that. Okay, so now I want you guys to connect this back to our traits of life. So think about the way that these different membrane characteristics might impact different traits of life. So order and structure. Remember, our membranes are organized in a very specific way, so they are structured, right? And they are involved in reproduction because the membrane has to expand in order for a cell to divide. It's definitely involved in sensing external stimuli, right? That was one thing I wanted to mention. So we go over to my little drawing over here. So if this is a kinase, right, the one that I'm most familiar with is histidine kinase 33 in the DSPA strain of Seneca cystis, species PCC6803. That's what I did my dissertation on. So these histidine kinases have these loops at the top, and then they have the tails, and between that is the membrane, right? And so if it gets very, very cold, the membrane is going to push inward, and then these loops change shape just a little bit, right? And when they do that, it changes the shape of the inside. And when the whole shape of the inside changes, then we get our signal cascade. So the membrane fluidity that is changed by temperature is responsible for this protein sensing the temperature. And then that protein sends its signal to the rest of the cell saying, hey, it's pretty cold, we better adjust to this. So that's how membrane fluidity is involved in cell sensing. Okay, and then our last thing is selective permeability. This is probably one you're pretty familiar with as well. So we got our inner leaflet and our outer leaflet, and in between we have a hydrophobic zone. And I mean, you have probably seen diagrams like this before. So polar molecules are not going to be able to get through that hydrophobic zone very easily. Non-polar molecules are going to be able to pass through. Smaller molecules are going to be able to pass through more easily than larger molecules. And charged molecules like ions will not be able to pass at all through that hydrophobic zone. Okay, so then this cell active permeability impacts the amount of energy that's required for different movements. So no energy is required for things that can pass through on their own, or things that are going through a channel protein like this one, or a facilitated diffusion assistant protein. Where we do see active transport is with ions. So you did this in your lab exercise. Ions get pumped against their concentration gradients in order for us to be able to do certain cellular energy generating activities, right? And so it takes energy to be able to move things against their concentration gradient. So if we're looking at this diagram right here, we have ATP synthase. Our hydrogens are going through ATP synthase from high to low, right? From high to low. So think about that. And then over here in our proteins over here, the ones marked B and C, we are pumping hydrogens against their concentration gradient. So this is the low region, this is the high region, and these proteins are using energy molecules to pump hydrogens into that higher concentration space. So looking at this, go ahead and tap the letter of which option is going to produce energy rather than consume energy. Okay, and then there's supposed to be a video here. It should be visible in the slides when you have them on Blackboard, but just take a look at that little video of what the cell membrane does during cell division, and then think about which ones of these options might be at play during cell division. Okay, and that is, oh, there's the video. But that is our last slide, and I hope that helped. If you have any questions, let me know.